Hello, ladies and gentlemen, and welcome to today's broadcast on technological innovations to speed protein characterization by Harsha Gunawardena, PhD. I am Jennifer Woods of LabRoots, and I will be moderating today's event. Before we begin, I'd like to encourage you to engage with us. You can submit as many questions as you want at any time you want during the presentation. Simply type them into the Ask a Question box on the left and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right, or use the Ask a Question box on the left. Please welcome Dr. Gunawardena.
Good morning. Welcome, everyone. First, I would like to give my sincere appreciation to Agilent Technologies for giving me this opportunity to present at today's webinar. In today's presentation, I plan to share a few important approaches we have developed in our laboratories or in collaboration with our partners to improve protein characterization workflows. As the title of my talk indicates, we are focusing on innovation to speed up protein characterization. Here is an outline that will guide you to my talk. First, I will give a brief introduction to my group's role in large molecule early development. Next, I will describe several different approaches that are beneficial to increasing biologics characterization and how these can be used in a stage-appropriate manner throughout the development of biologics. Finally, I will conclude my talk by summarizing our findings and what we hope to accomplish in the future. This figure represents the general stages in large molecule early development. Once a target is declared during pre-portfolio research, candidates in the range of 100 to 1,000 molecules get screened and filtered through lead generation and lead selection to less than a few dozen molecules that enters our department, the Cell and Developability Sciences, or CDS for short. The CDS group is responsible for the selection of the cell line that goes into late-stage development, and it's a function that sits in between discovery and development. The analytical function within CDS uses mass spectrometry to characterize molecules generated during cell line selection activities. These molecules include, as you can see, mAbs, drug conjugates, vaccine, and multi-specific mAbs used in immuno-oncology. Cell line development is depicted in this figure as a funneling process where the most desirable clone is selected based on product quality, expression stability, and titer. CLD starts with the transfection of the expression vector with a gene of interest into a host cell line, usually a Chinese hamster ovarian cell line. Single cells are next plated by isolating clones from pools using process known as VIPS, or verified in situ plate seeding. These single cell clones are then expanded to 96 deep well plate screenings, fed batch shake flask screenings, and final amber bioreactor screenings to finally select the top clone. The entire process takes 6 months from transfection to selecting a manufacturing cell. We use fit-for-purpose analytical methods, including mass spectrometric assays for product quality analysis throughout this screening process, as highlighted in the yellow boxes. Recently, a new process has been implemented in CLD to reduce timelines, where the process takes 4.5 months from transfection to clone selection. In the present paradigm, more molecules per target are transfected with fewer clone screenings, and early development starts from transfected pools rather than screened clones. The idea of this new workflow is to know more about the molecules faster and have backups ready to mitigate development risk. In order to meet these new deliverables, Analytical needs to innovate in technologies that can increase speed. We believe that automated solutions and other methods will reduce bottlenecks in the process, where CLD campaigns can move programs faster to IND. This brings me to the main topic of this talk. How do we accelerate biologics characterization? We in CLD are taking a multi-pronged approach with innovation happening both within the group and outside. These are the areas of interest that I will share today. Among these, we have established an automated data analysis as a platform method, while the others listed here are active research interests to accelerate biologics characterization. We have an end-to-end automated data analysis pipeline developed using Protein Matrix BIOS software. Key features of this software includes data generated from different MS instruments are automatically subjected to a vendor-agnostic bionic search, and these are done on group peptide MS data. Then extensive product variant searches of known modifications, as well as wildcard searches of unknown modifications are carried out. Validation of results and automatic export of results are also possible. Finally, data aggregation with molecular information is done using gene data biologics. Recently, we have integrated intact MS analysis workflows with peptide mapping for joint reporting and comparison of modifications. Here's an example of this approach where we compare modifications of an intact antibody after mass deconvolution with the reconstructed spectrum of modifications estimated from the peptide map of the same antibody. These profiles show good similarity. We are also interested in improving sample digestion speeds. Microdroplet-assisted digestion work was a Janssen collaboration with groups at the New Jersey Institute of Technology and Stanford University who are pioneering these methods. Conventional digestion methods are time-consuming and not amenable to high-throughput characterization of biotherapeutics. Here we describe a method of generating microdroplets using a mixing tea where reactants are mixed prior to electrospray ionization, or reactants are introduced separately and allowed to react after electrospray ionization. These methods enable comprehensive digestion and reduction of disulfides of antibodies at an unprecedented speed of 4 seconds in the former case to as fast as 250 milliseconds per sample in the latter case. Ides and PNGAse F, two of the most widely used enzymes for antibody digestion, in conjunction with a TSEP chemical reduction is performed via multiple sequential reactions or in a single reaction step using these microdroplets. Here's a monoclonal antibody Fab and single-chain FC fragments that are generated by Ides enzyme and compared with in-solution digestion. The top spectrum shows completed digestion of the antibody to Fab and single-chain FC fragments in 4 seconds. The bottom spectrum shows the same intact antibody that remains undigested after 5 minutes of in-solution digestion. Considering typically in-solution digestion of a mAb with Ides takes approximately 30 minutes or longer to complete, microdroplet digestion offers a 450x improvement in speed. We next generate antibody subunits by reduction and Ides digestion. The reaction performed in a T-junction takes place in approximately 4 seconds. The deconvoluted mass spectrum shows light chain FD and the single chain FC fragments that are each approximately 25 kilodaltons. The single chain FC fragments show characteristic glycans. The same reaction when performed in real time using 2 emitters, with the second emitter needle is nebulizing charged solvent ions that react with the neutral antibody fragments, generate charged fragment ions. All 3 antibody fragments are observed in 250 milliseconds, which is a 7 million x increase in digestion speeds. Ides digestion plays an important role in the structural characterization of antibody chains for modifications. The stop spectrum was generated as before by reduction and Ides digestion of antibodies that were subjected to glucose stress. The deconvoluted mass spectrum shows light chain, FD fragments, and their mono and diglycated forms. However, we are unable to determine FC glycation due to the presence of glycosylation. Both glycan and glycosylation masses are the same. We next performed PNGAse F deglycosylation in microdroplets, where 90% of the antibody gets stripped of their glycans in 4 seconds, as seen in the deconvoluted spectrum at the bottom. Finally, we combine PNGAse F reactions with Ides digestion and reduction to generate FC fragments free of any glycosylation. We are also interested in improving sample separation speeds to accelerate biologics characterization. I will show a couple of case studies on how we use separations technologies to speed up characterization. In the first case study, we are investigating clipping on an FC fusion protein. Clipping can take place where non-enzymatic mechanisms as well as due to the activity of enzymes present in cell culture harvest. Identification of clip sites are challenging, and clip sites are important in the development of safe and efficacious therapeutics. Here's our workflow for clip site identification. We label proteins with TMPP, which is a chemical tag that preferentially reacts with mostly N-termini of a protein, and then we digest this protein with trypsin or any other protease. The digested peptides that have the TMPP tag are combined by detecting of specific report ions upon MS2. Further report ions are used to trigger additional MS2 scans to confirm the sequence. Using this approach, we examine the diagnostic utility of report ions with over 1,000 TMPP labeled peptides from a K562 cell line total cell digest. In addition to the highly selective nature of these report ions, we noted that most labeling occurs at the N-terminus and that labeled peptides are significantly hydrophobic compared to their unlabeled peptide counterparts. Therefore, these peptides elute during the rapid sawtooth ramps of the LC gradient, as shown here. Using this information, we have completely automated the process for looking at clip sites where an assay map Bravo system is used for labeling and digestion steps of this workflow. And rapid LC separation followed by targeted MS is performed to detect clip peptides all within 12 minutes. Rapid and highly selective separations are achieved for detecting clipping of the FC fusion proteins, as well as NIS-MAP N-terminus, and we can obtain ROC curves with high sensitivity and specificity for the K562 label peptides when retention time of the label peptide used as the predictor. As you can see, we get a very decent ROC curve with the AUC of greater than 99%. In the second case study, we are using ion mobility separations performed using structures for lossless ion manipulations, or SLIM, to speed up biologics characterization. SLIM experiments were performed in a Janssen collaboration with Mobile Ion Systems, who is the exclusive licensee of the SLIM technology for commercialization purposes. In this workflow shown here, we investigate the released glycans from human serum IgG of lupus and healthy subjects. IgG from serum is purified, and glycans from purified IgG are released and labeled. These released glycan samples are then introduced using an Agilent LC by flow injection to a SLIM device that is coupled to an Agilent 6545XT mass spectrometer. The animation shows the operation of SLIM-IMS-MS system. We introduce a 30 centimeter long slug of sample by flow injection. These molecules are ionized by electrospray into a heated capillary and focused using an ion funnel. The total SIM path is 13 meters long, serpentine path where ions can be accumulated on board and released based on the collision cross sections. The arrival time for ions are recorded along with their mass spectra to give us a two-dimensional plot of mobility and MOZ. 55 ion packets are collected within these 2 minutes of flow injection runs. This gives us roughly 14 ion packets of data for the 30 seconds of the flow injection. This is a comparison of HILIC-MS with SLIM-IMS-MS analysis of released glycans. we can see that a typical HILIC run takes 15 minutes to separate glycans, whereas IMS can separate glycans in less than seconds of runtime. The table shows that many more glycan species are detected using 2 minutes flow injection run in comparison to the 15 minutes of HILIC run times. We detected glycans that have a wide range of MOZs and drift times. If we zoom into the most relevant glycans, we detect isomeric glycans for G0F, G1F, and the silated G1F, which is G1FS. Here's an overall view of the glycan distributions for both lupus and healthy subjects. We also have 2 global internal standards from pool glycans for healthy and lupus samples we collected, which are labeled as control 1 and control 2. It is known that galactose levels are implicated in autoimmune disorders. Low galactose levels have been reported for rheumatoid arthritis by us and others in previous studies. However, glycans derived from autoantibodies in our analysis for lupus does not show a statistically significant lowering of galactose when we examine the intensity ratios between G1F and G0F, that is a measure of a single galactose, or intensity ratios between G2F and G0F, which is a measure of 2 galactose units. We next looked at additional isomers observed by SIM separations. For example, G1F glycan is resolved into 3 reproducible peaks. This was performed in triplicate measurements for all samples that were analyzed. We used the intensities of the 3 G1F isomeric species as separately as predictors to categorize lupus and healthy subjects. To our surprise, rock curves of the 3rd G1F glycan species is more predictive of the disease state compared to the 1st and 2nd G1F glycan isomers. In the 3rd case study, we are using SLIM separations to speed up characterization of antibody-drug conjugates, or ADCs. An important parameter of evaluating the conjugation of a small molecule drug to the antibody is drug-to-antibody ratio, or DAR value. Traditionally, we have obtained DAR values using time-consuming 2D LC-MS analysis. Here we are using size exclusion chromatography separation in the 1st dimension, followed by reverse phase separation in the 2nd dimension, using multiple hard-cutting 2-dimensional LCs to obtain DAR values by the MS signal. In this analysis, we observed that the SEC retention time correlates very well with the DAR value. Alternative to this, we can speed up the characterization using SLIM. SLIM ADC experiments were performed in a Janssen collaboration with the Pacific Northwest National Lab. In this experiment, we were able to rapidly and simultaneously identify conjugated species and estimate the DAR values accurately for both the heavy and light chains of a model antibody-drug conjugate. These DAR values were obtained in significantly less time compared to size exclusion chromatography of the intact antibody. And we can also see that the DAR values compare quite well with the SEC-MS. Multiplexing. Multiplex sample analysis is another important method to increase throughput in biologics characterization. Isobaric labeling reagents such as tandem mass tags, or TMT for short, and other reagents have been used widely in proteomics for relative and absolute quantification. However, their use in pharmaceutical characterization is rather limited. Here we are using tandem mass tags to create isobaric labeled peptides to estimate the occupancy of a small molecule drug conjugated to antibody. As shown in the diagram, the workflow starts with labeling the unconjugated cysteine site of a protein of the ADC sample, and the same antibody without the drug we call a mock sample is then labeled with a first pair of cysteine reactive TMT reagents, or iodo-TMTs. These parallel samples are then trypsinized, and the resulting peptides are labeled with a second TMT tag that are reactive to the N-terminus and the lysanes of the peptides. The peptides are pooled and then mixed in equimolar ratios and analyzed by LC-MS. The peptides, when subjected to MS2, generate these TMT report ions as shown in the spectrum for both the first and the second pairs of TMT labels, from which the occupancy of the drug conjugation can be derived as shown in the equation below. Here are a few examples of site-specific occupancies estimated for cysteine conjugate. Different sites have different occupancies as seen in the top panel. The spectra show the reporters, and the bottom panel shows the corresponding fragment ions that localize the conjugation sites. Once we obtain the occupancies for each site, we make use of the non-cysteine TMT ratios of the spectra, which have exclusively the second TMT label, to obtain a normalization factor between the conjugate and the mock sample. We then use this factor to correct occupancy estimates for each site-specific cysteine conjugation site. Using this approach, we can multiplex up to 4 different conjugation reactions to estimate occupancies of a drug in a single experiment. The experiment shown here is a site-specific occupancy estimation using TMT 10-plex reagents to monitor the conjugation of 4 different conjugation reactions in a single run with unprecedented throughput. Our final approach to speed up biologics characterization is rapid sample introduction with no separations. This type of analysis is particularly important for multi-specific antibodies that can be produced to desired heterodimers as well as undesirable homodimers during single-cell cloning. Cell line development activities require rapid product quality analysis, specifically looking at chain pairings of multi-specifics. One needs to screen large number of clones during cell line development stages to identify and rank order cell lines with the highest percentage of these heterodimers. We are using RapidFire-MS with cycle times of 8 to 10 seconds per sample to speed up the processing of large volumes of samples. Our approach starts with purified cell culture material and subjecting samples mostly in 96 well-formed to RapidFire-MS analysis. The data generated from these experiments can be split to individual files and processed using our intact MS data analysis pipeline, where intact mass spectra are deconvoluted and intensities are reported for each clone for various species, as shown in the heat map at the bottom. Here's an example where only 13% of the high titer clones moved into cell line selection due to high levels of homodimers and half-mers detected through this type of analysis. In addition to chain mispairing, we are also exploring the use of RapidFire-MS for other applications. Product quality attributes such as glycosylation can be estimated by performing RapidFire-MS analysis on antibody fragments. In this example, we investigate the glycans of the heavy chain located in the Fab and FC domains using Ides. We can generate the FD and FC regions of the antibody and analyze each individual chain for their glycan distributions. In addition, glycosylation on multi-specifics can be compared by reduced mass analysis. The non-identical glycans on each heavy chain can be rapidly analyzed using RapidFire during product quality screening. We have compared glycans between LC-MS and RapidFire-MS approach and find a good correlation between the major glycoforms. Finally, RapidFire-MS can be used to examine mAbs under native conditions. And here we are using charge reduction to strip charges on a monoclonal antibody to increase the distribution of peaks that we believe will help to improve charge deconvolution. In this example, we are increasing the flow of the charge reducing reagent introduced from the Agilent reference spray every 5th injection during the rapid fire run. In conclusion, I hope you have seen the variety of methods we have developed to accelerate biologics characterization in a stage-appropriate manner. These enabling technologies include increasing digestion speeds several million-fold using microdroplets. As I pointed out, microdroplets accelerate otherwise very slow reactions and is transformative in the characterization of biologics with increased throughput and versatility. Next, we examine short gradient LCs and ion mobility separations to speed up analysis times at unprecedented speeds. Sample multiplexing for accurate and fast quantitation was also demonstrated using tandem mass tag approaches. And finally, rapid sample introduction from screening to product quality assessment of molecules was demonstrated. What is the future? Our future is looking bright. We are continuously innovating and improving our analytical methods, informatics capabilities to become an industry leader in biologics characterization. We also see opportunities for improvement in sample management and data tracking systems. Integrating high throughput mass spectrometry with robotic sample preparation is one of the areas that we are looking into. Also, increasing the information content of our analysis by integrating various assays, mass spectrometry assays with probably biophysical assays in an autonomous fashion so that we can integrate results and deliver these results to our stakeholders in an integrated fashion. We envision technological innovation will provide time to value in biologics characterization. With that, I would like to thank all the contributors at Janssen and our research partners and collaborators. Finally, I would like to thank you for your participation in this webinar and happy to answer any questions. Thank you.
Thank you, Dr. Gunawardena, for your informative presentation. We've received some great questions already. Our first question is microdroplet digestion being performed on chemical reactions?
It is. So this technique has been pioneered by groups both at Purdue University and at Stanford. And both these groups, Professor Cooks and Professor Zaire, have performed these types of reactions for organic molecules. And this has a huge potential even for pharmaceuticals, biologics, where certain biologics need to be modified. One such example, which I've shown, is antibody-drug conjugates and various types of chemical modifications like TMPP. And these are some of these chemicals that we can use to modify the proteins, antibodies and antigens. And these are some areas that we are exploring in the future.
Okay, great. Thank you. I have another question. Is the multiplex experiment, in the multiplex experiment, what is the use of the second TMT tag?
Yeah, that's an excellent question. So this multiplex experiment is a little different to the typical proteomics experiment where researchers typically use one type of labeling reagent, either it's lysine or primary amine reactive reagents or cysteine reacting reagents. It has never been used together in a single experiment. So the idea behind this type of design is to use the second label to do normalization, especially when we are looking into conjugates in this particular experiment where we're trying to determine occupancy. The two work streams, parallel work streams, needs to be the protein amounts in the two parallel workflows need to be the same, similar. So in order to account for any errors introduced during experiments, such as mixing errors and reaction errors, we have to correct for the differences. So we do that by using the second reagent, which typically reacts with the N-terminus or a lysine on the peptide, that we can use to normalize the ratios and correct for any errors that were introduced during sample preparation.
Okay, thank you for that. So far, we have one more question. In the clipping experiment, are there any tags you can use for detecting C-terminus clip sites?
Yeah, excellent question again. So far, the reagents that are available chemically is to react with the N-terminus. There's no reagent that can efficiently tag at the C-terminus. However, as I pointed out, clipping is pretty important attribute in biotherapeutics characterization. Not all peptides that we measure during such experiments create peptides that are sufficiently long to be detected only at the N-terminus. So there needs to be reagents that can also attach to the C-terminus. Although there aren't any reagents that are available in the market, at least for now, that reacting with the C-terminus, there's some research groups, especially in the organic synthesis, where people are trying to develop C-terminal probes, bioconjugation probes, by using photochemical reactions. So these are still research interests, and they're still in research and not commercially available. But that is something that we would like to see happen and made available to the broader community, biological community, so that we can use this for biologics characterization.
Right. Well, thank you so much. As a reminder to our audience, questions we did not have time for today and those submitted during the on-demand period will be addressed via the email address you provided at the time of the registration. Thank you again to our speaker, Harsha Gunawardena, PhD, and to everyone who participated in this presentation. We hope you enjoy the rest of the event.